There is a parasite living inside the brains of approximately 2 billion people right now. It produces no symptoms. It causes no pain. Most of the people carrying it will never know it's there. It arrived quietly, through undercooked meat, through contaminated water, through the litter box of a domestic cat. It crossed the blood-brain barrier, formed microscopic cysts in neural tissue, and settled in. And then it started making changes. Not dramatic changes, not the kind you would notice. Small adjustments to dopamine levels, subtle shifts in risk tolerance, gradual modifications to the way fear registers in the brain, changes so minor, so incremental, that the person experiencing them interprets them as their own personality. Scientists have spent decades studying what Toxoplasma Gondii does to the animals it infects. What they found should concern you, not because of what it does to rats, not because of what it does to chimpanzees, but because of what the evidence suggests it may already be doing to you. The question is not whether this parasite is dangerous. The question is whether you would even know if it had already changed you. Toxoplasma Gondii is a single-celled parasite. Under a microscope, it looks almost elegant, crescent-shaped, translucent, smaller than a human red blood cell. It has been on this planet for millions of years. It has infected virtually every warm-blooded species we have studied. And it has spent all of that time perfecting a single strategy, getting inside a brain and rewriting what happens there. To understand what it does, you first need to understand what it wants. Toxoplasma can reproduce sexually in only one type of host, members of the cat family: domestic cats, wild cats, lions, leopards, cheetahs, any felid. When Toxoplasma reaches the intestinal lining of a cat, it undergoes sexual reproduction, produces oocysts, microscopic eggs, and those oocysts are shed in the cat's feces. From there, they can survive in soil and water for up to 18 months, resistant to most disinfectants, invisible to the naked eye, waiting. When another warm-blooded animal ingests those oocysts through contaminated food, through water, through contact with infected soil, the parasite awakens. It crosses the intestinal wall, enters the bloodstream, and begins moving toward its target, the brain. Once inside neural tissue, it forms cysts, microscopic clusters of dormant parasite cells embedded in brain matter. In a healthy immune system, the body suppresses the infection and keeps it contained. The cysts remain. They don't disappear, they persist, potentially for the lifetime of the host, quietly present in tissue that controls thought, emotion, and behavior. This is not an accident. This is the strategy. The clearest evidence of what Toxoplasma actually does comes from rodents. The intermediate host, the parasite, evolved to manipulate most effectively. A mouse infected with Toxoplasma loses something specific, not its general awareness, not its ability to move or eat or navigate a maze. It loses its fear of cats. Not a vague reduction in anxiety, not a generalized blunting of the stress response. It loses, with remarkable precision, the aversion to cat odor, the deep evolutionary instinct that has kept small rodents alive around feline predators for millions of years. In controlled laboratory studies, infected mice and rats don't simply fail to avoid cat urine. They approach it. Some show responses more consistent with attraction than aversion. The parasite has reached into the brain of a small animal and deleted the one instinct most likely to prevent it from reaching a cat. The mechanism is now partially understood. Toxoplasma cysts concentrate in the amygdala, the region of the brain responsible for processing fear and threat response. The parasite encodes an enzyme called tyrosine hydroxylase, which is a precursor to dopamine synthesis. By increasing dopamine in specific neural circuits, the parasite suppresses fear-related signaling in a targeted way. It doesn't sedate the animal. It doesn't impair its cognition. It modifies one very specific response. the one that stands between the parasite and its destination. The mouse walks toward the cat. The cat eats the mouse. The parasite completes its life cycle. This is not random mutation. This is not coincidence. What Toxoplasma does to a rodent's brain is one of the most precise examples of behavioral manipulation in all of parasitology. In 2016, researchers from the CNRS in France and Germany published a study in the journal Current Biology that extended these findings to our closest living relative. They tested chimpanzees, both infected and uninfected with Toxoplasma, by exposing them to the urine of different large predators, leopards, lions, and tigers. Uninfected chimpanzees displayed the expected response to leopard urine, their only natural predator in the rainforest environments they inhabit. They avoided it. They showed stress-related behaviors. The instinct was intact. Infected chimpanzees showed something different. They lost their aversion to leopard urine specifically, not to lion urine, not to tiger urine. Neither of those animals is a natural predator of chimpanzees in the wild, only to leopard urine, the scent of the one animal that actually hunts them. Did the infected individuals show a diminished fear response? The parasite, operating inside the brain of one of the most cognitively sophisticated animals on Earth, had modified fear response with the same precision it uses in rodents: targeted, specific, functional. The researchers noted something else. The behavioral modification in chimpanzees may be a residual manipulation trait, one that evolved when our own ancestors were still being hunted by large felines. Modern humans are no longer regularly preyed upon by cats. But the parasite's machinery is still there, still running, still making adjustments in brains that no longer need to worry about leopards. What it's adjusting instead is something researchers are still working to understand. The human data is more complex. Establishing direct causation in human behavior is difficult. People are not laboratory mice, and the interactions between infection, genetics, environment, and behavior involve layers of variables that controlled studies struggle to isolate. The scientific community is appropriately cautious about strong causal claims, but the correlations are consistent, and they have been replicated across populations, across continents, and across decades of independent research. Jaroslav Fleger, a Czech biologist who has spent much of his career studying Toxoplasma in humans, conducted a series of studies beginning in the 1990s that compared the behavior and personality profiles of infected and uninfected individuals. What he found was not dramatic, but it was measurable. Infected individuals showed longer reaction times on cognitive performance tests, a difference modest in isolation, but consistent across multiple studies involving hundreds of subjects. A follow-up study collected blood samples from 146 people who had been identified as responsible for causing traffic accidents, either as drivers or pedestrians, and compared them with 446 control samples from the general population. The seroprevalence of Toxoplasma infection was significantly higher in the accident group. Slower reaction times, reduced risk perception, a subtle impairment in the processing that keeps people out of danger. The same pattern the parasite uses in mice, targeted modification of the fear and caution systems, showing up faintly but measurably in human populations. A meta-analysis of 38 independent studies, published through Oxford University Press, found that Toxoplasma infection increases the odds of developing schizophrenia by a factor of 2.7. This does not mean infection causes schizophrenia in most people. The majority of infected individuals show no psychiatric symptoms. But the statistical relationship is one of the strongest documented links between any infectious agent and a major psychiatric disorder in the scientific literature. Additional studies have documented associations between Toxoplasma infection and major depressive disorder, bipolar disorder, suicidal behavior, and impulsivity. In 2017, a study published in the Proceedings of the Royal Society found that entrepreneurs and business founders tested positive for Toxoplasma at significantly higher rates than non-entrepreneurs, and were more likely to have started businesses in high-risk industries. The infection was associated, statistically, with greater tolerance for financial and professional risk. None of these studies establish that Toxoplasma is directly causing these outcomes in every infected person. What they establish is a pattern, a consistent skew in behavior and risk tolerance across populations in the direction the parasite has been shown to push cognition in every other species it infects. The scale of the infection is what makes this impossible to dismiss. Toxoplasma gondii infects approximately 1/3 of the global human population. That estimate, drawn from serological studies across dozens of countries, represents roughly 2 billion people currently carrying dormant cysts in their neural tissue. Prevalence varies significantly by region, around 10 to 15% in the United States and parts of Northern Europe, rising to over 60% in parts of South America, Eastern Europe, and Sub-Saharan Africa. The primary transmission routes in humans are well documented by the CDC. Ingestion of undercooked or raw meat containing tissue cysts, particularly pork, lamb, and venison. Contact with soil or water contaminated with cat feces containing oocysts. Unwashed produce grown in contaminated soil. Congenital transmission from a mother infected during pregnancy, which can cause severe neurological damage or death in the developing fetus. A single infected cat can shed up to 500 million oocysts in its feces during the first weeks of infection. Those oocysts survive in the environment for over a year. They are resistant to chlorine-based disinfectants at concentrations used in municipal water treatment. They can pass through standard water filtration in some conditions. You cannot smell them. You cannot taste them. There is no routine screening for Toxoplasma in healthy adults in most countries. The standard medical position is that infection is asymptomatic in immunocompetent individuals, and for most people, most of the time, that is correct. The acute phase passes without incident. The cysts form. Life continues. Normal, as far as anyone can tell. What science still cannot answer, and what makes Toxoplasma uniquely unsettling among all the organisms discussed on this channel, is the question of degree. Every other animal covered here operates externally. You can see a chimpanzee, you can avoid a grizzly bear, you can stay out of the ocean to avoid an orca. The danger is outside you, and the calculus of survival, however difficult, involves a boundary you can perceive. Toxoplasma eliminates that boundary. If it has infected you, it is already inside the structure that generates your thoughts, your fears, your decisions about risk. The cysts in your amygdala are not passive. They are metabolically active. They are producing enzymes that interact with your dopamine system, and the behavioral changes they may be producing are, by definition, invisible to the person experiencing them, because those changes are being processed as normal cognition. The mouse approaching the cat does not know it has been changed. It is simply, from its own perspective, less afraid than it used to be. Researchers have noted a disturbing implication of the chimpanzee study. If the parasite's manipulation in humans is an ancestral legacy of a time when our ancestors were hunted by large felines, then the specific fear it is suppressing in modern humans may no longer serve its original purpose. The machinery is running. The target of that machinery, in a world without leopards outside our windows, is unclear. What we know is this: in every species studied, Toxoplasma gondii modifies the host's relationship to risk and fear in a direction that benefits the parasite. The modifications are subtle, they are consistent, and they scale with the number of cysts. The longer the infection persists, the more embedded the changes become. The cysts, once formed, do not dissolve. There is no treatment that eliminates them from brain tissue. Antiparasitic drugs can suppress active infection, but the dormant cysts in neural tissue persist through standard treatment. In most people, in most circumstances, the immune system keeps them contained, but they are there. In approximately 2 billion people walking the planet right now, going about their lives, making decisions about what risks are worth taking, what fears are reasonable, what impulses to follow, There are dormant clusters of a parasite that has spent millions of years learning exactly how to push those calculations in a specific direction. You can be afraid of what you can see. That fear has a name, it has a shape, it stays outside you. What Toxoplasma does is something different. It gets inside the system that generates fear itself. And from there, quietly, without symptoms, without pain, without any signal you would recognize as an intrusion. It decides how afraid you are allowed to be. The question is not whether this parasite is dangerous. The evidence on that is clear. The question, the one you cannot answer without a blood test you have probably never had, is whether it is already in you. And whether the version of you reading this right now, weighing that possibility, deciding how seriously to take it, is doing so with a brain that is entirely your own.